Okay, it's my pleasure to introduce our next speakers. So Dr. Kyle Lowe is a Packard Fellow, a Pew Scholar, a Human Frontier Young Investigator, a Baxter Foundation Scholar, a Forbes 30 Under 30 awardee, a Genova. I looked all this stuff up.
Yes.
I mean, yeah.
Yes.
Yeah.
Yes.
Endowed scholar and assistant professor of developmental biology here at Stanford, and a great guy.
That's how you know he doesn't tell the truth that often.
And Kyle is working in stem cells and looking at ways of developing motor neurons. And I won't say anything more about what he's going to do. He's going to talk about. But I think we all know, those of us who study ALS or SMA, that not all motor neurons are the same, because we see people that have different motor neurons. Some get sick and others don't. And it's always been a mystery why. And hopefully Kyle's work is going to shed some light on that. So thank you very much for shedding some light here.
Yes.
Thank you.
Thank you, everyone. So today, I'm going to talk about creating human hindbrain motor neurons from pluripotent stem cells with the goal to understand how these debilitating bulbar symptoms arise that all of you are treating in your patients. So by way of introduction, hindbrain motor neurons reside in your brainstem, and they project through the cranial nerves to control facial, jaw, eye, tongue, and neck muscles. They're thus critical for eating and swallowing, as well as facial movements and speech. And as we've heard in the preceding sessions, these hindbrain motor neurons degenerate in ALS and SMA, leading to difficulties in speech and swallowing. And we've heard of the great advances clinically to try to manage these symptoms. And all of you in the audience know better than I do that difficulties in swallowing can be life-threatening because they can lead to choking and food aspiration. So our goal is to create a massive supply of human hindbrain motor neurons in a petri dish. And if we could really create an infinite supply of human hindbrain motor neurons, this would allow us to build better disease models, to test for new drugs, and maybe in the distant future to actually transplant these motor neurons to regenerate missing and dying ones. But where do we start from? We start from certain kind of stem cells in a petri dish known as human pluripotent stem cells, shown here on the left. And these stem cells have two special properties. First, they can be grown endlessly in a petri dish. And second, they can theoretically make any cell in the body. When I say that these embryonic stem cells can be grown infinitely, I really, really mean it. So an embryonic stem cell, although it's just really tiny, just 10 micrometers wide, it divides every 18 hours. So in the course of 1 year, a single embryonic stem cell today in our lab could make 10 to the 146 embryonic stem cells, which is such a vast number. It's vastly, vastly, vastly greater than the number of stars in the universe. So there's not enough money being generated at Stanford Children's Hospital, adult hospital, the country, or the world to even buy the media to grow this many cells. So we can really, really create infinite numbers of stem cells. But the big challenge for us is how can we create these hindbrain motor neurons from them? And this then in turn raises the question, what are all the steps through which a stem cell becomes a hindbrain cell? And here we came across a bit of a surprise. So all of you know that we often think of the brain as a single organ, right? It's pretty obvious. And it's thought to come from a single common progenitor in development. The term here is called the neural ectoderm, but the exact term isn't that important. And that really makes sense, right? If you think about the liver or the pancreas, we think there's a single liver stem cell or a single pancreas stem cell that constructs the entire organ. But we found a big surprise that the brain isn't actually one organ, but really a composite coming from two totally different sources. One that forms the forebrain and midbrain in red, and the second source that forms the hindbrain in green. We think of this new view that the brain isn't one organ, but actually two mashed together, has very broad implications for development, evolution, and disease, as I'll talk about today. I'm going to focus specifically on the hindbrain for the sake of bulbar symptoms. So first, I want to thank our stellar lab members who led this project, including graduate students Carolyn Dundas and Ryan Jokai, as well as several research professionals, Rachel Kang, Hadia Hassan, and Rachel Solomon. So we came into this actually completely by accident. So there's been a lot of efforts to convert embryonic stem cells into brain cells. But we noticed that these methods tended to make forebrain and midbrain cells. And it was a bit harder to make hindbrain cells. We thought, that's kind of weird. We didn't really know why. So we took a step back to the drawing board to look at the classical model of developmental biology of how the brain emerges. So in developmental biology, so when you're an embryo, so in a human, this would be maybe you're 2 weeks old, mom doesn't even know she's pregnant yet, a pluripotent cell becomes a cell type known as the ectoderm. So the ectoderm, mesoderm, and endoderm, the ectoderm are the outermost cells, and the ectoderm becomes a certain cell type called the neural ectoderm. So neural, brain, ectoderm, ectoderm. So this is thought to give rise to the entire brain, the forebrain, the midbrain, the hindbrain. And this model was first proposed by a scientist called Peter Newcoop, a Dutch embryologist in 1952. And in fact, entire books have been written about this cell that can theoretically create the entire brain, including a book by Lewis Thomas. So he was the president of Memorial Sloan Kettering Cancer Center, and he won the National Book Award for a book called Medusa and the Snail. He wrote very beautifully. You start out as a single cell that becomes 2, 4, and 8. Then at a certain stage, there emerges a single cell that can give rise to the entire human brain. The mere existence of that cell should be one of the great astonishments of the earth, which I agree, right? It's kind of amazing that the entire brain can be constructed from just a simple clump of cells. But we found it was a bit more complicated, and there was not one, but 2 different brain progenitors. One in red that makes the forebrain and midbrain, and the other in green that makes the hindbrain. And I'll provide the evidence that supports this model in a moment. But when we came across this, it immediately clarified why it was much easier to turn stem cells into the forebrain and midbrain, and it was harder to make the hindbrain. Because the classical methods to make brain cells really and more accurately create this red progenitor cell that can make a forebrain and midbrain. And if you didn't really know that the hindbrain comes from a totally different progenitor cell than the rest of the brain, you'd never really know to make that progenitor cell in the first place. So the analogy I like to use is this is like navigating down a main road leading to the forebrain and midbrain. And if you didn't know there's a concealed side entrance leading to the hindbrain, you wouldn't really know when to turn off your drive home to get there. So how did we come across this idea that there's 2 different brain progenitors? Because this idea is actually very deeply counterintuitive in my field, and we didn't actually believe it ourselves for quite a long time. So my graduate student, Carolyn, simply just looked. So Carolyn looked at a mouse embryo that's 7.5 days old. So gestation in a mouse is roughly 20 days, so 7.5 days is pretty early in its life. And this embryo has just undergone gastrulation, so it's made the endoderm, the mesoderm, and the ectoderm. So in fact, it's so early that there's no head and there's no tail. It just looks like this big cup, basically. But in this cup, it has a front and a back. And what Carolyn is doing here is that they're staining for the expression of 2 genes. One in red is called OTX2. The name isn't that important to just keep in the back of your mind that this marks the first brain progenitor. So I'm going to call this the anterior neural ectoderm, or the ANE. And Carolyn found that there's a second gene in green, which is called GBX2, which marks the posterior neural ectoderm, or the PNE. So just to take a bit of a step back, at this point in development, all the outside cells in the embryo are thought to be these ectoderm cells. And we really believe that there would only be one kind of ectoderm cell. But what Carolyn here is showing is that there's not one kind of ectoderm cell, but there's really 2 of them that express different kinds of genes. So for us, this is a big surprise because we thought, why should there be 2 kinds of brain progenitors coexisting? There should really only be one at this stage. So Carolyn then went on to ask, are these brain progenitors actually functionally different? Do they give rise to different kinds of brain regions? So what Carolyn used is an elegant technique called lineage tracing. And simply put, in lineage tracing, you can label a cell red, and that is in its DNA. So whenever the cell becomes another kind of cell, migrates, or does anything, it's always going to remain red for the rest of its life. And we thought, if we label the second pool of brain progenitors, it gives rise to the whole brain, this would invalidate our idea. But this second brain progenitor only makes the hindbrain, so only the hindbrain is red, this would support our idea. So now Carolyn is looking at a mouse embryo at a bit later in its life, and you can see that although it doesn't look like a human, it's looking quite a bit closer with the head on the right and the tail on the left. So the brain is on the right, and you're seeing a zoom in, and you can see the forebrain, the midbrain, and the hindbrain. But when Carolyn did this so-called lineage tracing, they found that those second type of brain progenitor in red only ended up in the hindbrain, but not the forebrain and midbrain. I know that this is a really simple image, but I think what was really amazing to me is if you just took away the colors, it would just all be gray, right? The brain is just gray. But if you could look back in developmental time with us, you could really see that the back half of the brain comes from a totally different cell source than the front half. So this was really a stunning moment for us. And we realized that the brain, although it appears as one organ, you can think of like a lake. It's like a single placid body of water. A lake can nevertheless be formed from the convergence of two upstream streams that merge together if you just could look a bit more upstream. So having now discovered that there are these two different kinds of brain progenitors, we then realized that in a petri dish, this would totally change the way that we'd go about making these brain cells, because now we have to make these two different kinds of brain progenitors. And having that discovered that this posterior neuroectoderm in green makes the hindbrain, we could then force embryonic stem cells to turn right and only become the second kind of brain progenitor, and then to make hindbrain neurons. And we're really happy to work very closely with John Day and the Neuromuscular team to really guide these experiments. So we've now succeeded in making human hindbrain motor neurons in a petri dish from stem cells. So I'm showing you here is immunostaining of hindbrain neurons. So you know that neurons have these beautiful long projections that they use to communicate. And we were really struck to find that in a petri dish, these human hindbrain motor neurons also had these beautiful long projections in cyan. They express many genes that we expect of hindbrain motor neurons. And importantly, they're electrically active. So here I'm showing you a live movie of these human hindbrain motor neurons firing. So green indicates a spike in calcium, which is indicative of electrical activity in these neurons. So this is really a big moment for us because this was, to our knowledge, the first time that anyone had made human hindbrain motor neurons in a petri dish that were electrically active, which is what we expect of them. So finally, we then asked how long ago in evolution these two brain origins emerged, because as you know, in evolution, one of the biggest questions is where does the brain come from? Now, though our lab focuses on humans and mouse, which are both mammals, we found that these two brain progenitors were conserved even to chicken, to zebrafish, and finally to acorn worms. And the fact that these are found in acorn worms was really stunning to us because acorn worms are deuterostomes that diverged from us over 500 million years ago in evolution, so even before the dawn of chordates. So I'm showing you on the right a photo of an acorn worm, which I hope you'll agree looks nothing like one of your patients. I guess Mr. So-and-so is a 1-year-old presenting with so and so on. But if you could look in the embryo, it actually looks really similar to those mouse embryos I showed you. And on the left, it has this first brain progenitor expressing OTX in purple. And on right, it has the second brain progenitor expressing this GBX gene in yellow. So this really suggests that these two brain progenitors are evolutionarily really, really ancient. And for hundreds of millions of years, they have been conserved. And this also really makes a lot of sense, because although you may not do a bulbar function test on this acorn worm, my collaborator also has a paper in Nature that shows that molecularly, acorn worms, although they're very, very simple, they actually have a hindbrain, molecularly speaking. So in summary, in ALS and SMA, hindbrain motor neurons degenerate, thus leading to difficulties in speech and swallowing that we've heard a lot about today. We can now produce massive numbers of human hindbrain motor neurons in a petri dish from pluripotent stem cells. We hope that this ability will help us build better disease models for these bulbar symptoms that you see, to test for new drugs eventually, and finally to create a supply of human hindbrain neurons for transplantation one day into patients. And from the basic science side, it's often assumed that the brain is a single organ that comes from a common embryonic progenitor, but we find that the brain isn't really a single organ, but really a composite formed from two totally different progenitor sources, one for the forebrain midbrain and the other for the hindbrain. And we think that this has very, very broad evolutionary implications. So you take even a further step back from acorn worms to jellyfish, which are called cnidarians. They don't actually have one brain, but they have two physically separate nerve rings, one in the front of the body and one in the back of the body. And you can imagine that there's not many neuroscientists that study these jellyfish, but it's been proposed that the front nerve ring is maybe for sensation and the back half is for motor control, but it's really hard, obviously, to test or prove these things. So one completely speculative and untestable theory that my collaborators have forced us to remove from the paper is that all along, that God really had no plan to make one brain, but rather two physically separate nerve rings at different parts of the body from different progenitors that did different things. And then when it came to us, as you know, evolution rarely creates new things. It often just takes two existing things and pushes them together. In this case, these two pre-existing brain progenitors and pushing them together so they synaptically integrate more efficiently to give rise to the appearance of one brain, but really from dual origins. So we think that, actually the idea that the brain is one organ is a historical anachronism because scientists and doctors did dissections, obviously, of humans first. But if people had dissected jellyfish first, they'd obviously say, well, look, they have two nerve rings, and now we have one brain. So obviously, it just came from two things that came together. So in the last minute, I just want to touch on the spinal cord, because I know many of you in the neuromuscular field also think a lot about this. And now we're really curious that although the second brain progenitor makes the hindbrain, we now think there's actually a third independent progenitor that makes the spinal cord. So there's many, many different origins to the central nervous system. And as John alluded to, those of you who are clinically know much better than I do that SMA does not affect all motor neurons equally, although it's called a motor neuron degenerative disease. And that if you look at cervical, thoracic, lumbar, and sacral motor neurons, they really are affected to very differing extents. So in SMA, the diaphragm, which is in the cervical region, innervated by phrenic motor neurons in the cervical region, is largely spared, whereas by contrast, the thoracic motor neurons that innervate the intercostal muscles degenerate, leading to respiratory failure in these type 1 SMA patients. And to the best of my knowledge, as John said, we don't really understand why are some motor neurons affected and why are others spared. So we're thinking that just like in the brain, that these different brain regions have very, very different ancestries and ways of coming about, it might be the same thing for the spinal cord, and that these cervical and thoracic motor neurons, although you might think of them as really, really similar, they might actually come from totally different paths in developmental, and that this might lay down very deep differences between them that could affect their vulnerability to various diseases shortly after birth and in adulthood. So the next goal for our lab is to convert human pluripotent stem cells into cervical, thoracic, lumbar, and sacral specific motor neurons. And again, we'd have the same questions. Is there a common progenitor for the entire spinal cord? Could there be multiple progenitor cells? What are the signals that induce each of these spinal cord segments? And if we can create cervical and thoracic motor neurons in a petri dish, would one be more susceptible to SMA and not another? And if we can understand why some motor neuron subsets are more resilient to the disease, that would actually be really great, because that's an experiment of nature that we have mutations in the same gene that affects one motor neuron, but not a sister motor neuron located really nearby. If we could just learn how that happened, that could lead to better therapies. So in summary, I want to thank our stellar lab members, including Carolyn, Ryan, and Rachel, as well as our entire lab. And in particular, I really want to thank John Day, Tianyi Zhang, and the SMA Foundation for getting us into this in the first place and really teaching us about SMA and encouraging our interests, and support from various sources, and in particular the SMA Foundation. So thank you.
I was really happy to see you end with 3, because my first question was like, well, if there's 2, there's probably more. So
Yes.
it was really great. And then it may sound ignorant, but so the glow that you had, is that after the neural tube, right? It has to be, right? So
Before.
is the neural, it is before.
That's the surprise. So basically when the neural tube, so in developmental biology, we're taught that the ectoderm forms in gastrulation, then it rolls into a tube. By the time it rolls into a tube, by gene expression, you can distinguish the forebrain, the midbrain, the hindbrain. There's no question that when the neural tube forms, there's already differences, right? But I think the big surprise here is even before there's a neural tube, just as the ectoderm is forming, there's already very early differences. So these differences occur one step earlier than we thought that they did. Yeah.
Great, thanks,
Okay,
Kyle.
thanks.
I think, and,
I have a question too.
Connie.
Like you're walking over. First of all, that was awesome. A masterful presentation, very cool research.
you.
Thank you so much for sharing it with us. I'm interested in, thank you for telling us your next steps in this research. I'm very interested to see how that goes. I'm also interested in hearing about how do you what are plans on making it more disease specific, so that way we can look at different types of motor neuron disorders or beyond.
Yeah, that's right. That's a great question, and thanks for your kind words. I think the next step for us is that John Day's team, together with Joe Wu's team, has created from Stanford human-induced pluripotent stem cell lines from 2 patients with type 1 SMA. So these cells have the exact genetic mutation of these patients, and we've just now gotten approvals to convert those type 1 SMA stem cells compared to control stem cells into these motor neurons. So one hypothesis would be that if we could create cervical specific or thoracic specific motor neurons, that really in the SMA patients there should be more dysregulation or differences in splicing in the thoracic ones that are affected because the intercostal ones, as opposed to the phrenic ones. I think what's really fascinating about all of this is even the classification, as you know, of cervical, thoracic, lumbar, and sacral is grossly oversimplified. In the cervical region, there's phrenic motor neurons that innervate the diaphragm, there's arm innervating motor neurons, and John and I have had great conversations that there's massive differences between those. So our goal is really to create not just cervical specific motor neurons, but really phrenic specific or arm innervating specific motor neurons. So there's just gonna be a lot of fun, and I've told John we're not going to rest until we have every single motor neuron, and there'll be an institute dedicated to the John Day creation of every motor neuron subtype. Yeah.
Thank you for an amazing talk. It makes me so happy to meet
young people who are brilliant. It gives me hope.
Yes.
So can you give me some sense of the time frame for your project and how you, what was the time span from beginning to end when you determined that there?
Yeah, that's a great
have
question.
been
So the time span for every individual experiment is roughly 14 days. So from starting from an embryonic stem cell today, within 14 days, we could make these hindbrain motor neurons. So that's how long each individual thing takes. For this project, it's been a very, very long time in the making, I think roughly 8 years, and this paper is not yet published. In fact, we now have 4 reviewers that have assembled 96 comments for us to address, so that's what I'm doing right after this. But yeah, basically when we began this project, we really believed that there was a single kind of brain progenitor, and the big surprise was that his summer intern, who was just doing this, and we had no idea what was going on, and he tried in a petri dish that he could make 2 different brain progenitors, and I told him this is some mistake, because obviously he just started this lab like 1 month ago and so on. But really, I find what's really inspiring is that there's this college student who had no idea what was going on, who came across all of this. Then a grad student joined and said, that seems pretty interesting. I really want to work on this. And that's basically how it began. But it's just in many, many years. And the SMA focus has become more central the past few years now, given to know John and realizing that these bulbar symptoms are so debilitating and these hindbrain motor neurons are kind of a unique entry point into this. So it's been a long time in the making.